We're going to just do a very short unit here on genetic mutations, and then we're going to talk more about those a little later on in the year because DNA technology is putting us in a place where we can start to treat some genetic disorders that we never thought we'd be able to. So what we're going to do for this little short section here is I'm just going to talk about what a genetic mutation is and the different types of mutations. So it's just going to be a quick overview of those things. So let's get started here. So the term mutation you've heard before, right? You've watched all these X-Men movies and, DC Comics movies, and you've heard of mutants before. Well, those are different kinds of mutants, right? So if you get exposed to like a massive dose of radiation, chances are you're not going to be able to fly. You're probably not going to be able to shoot webs. You're probably going to have cancer, unfortunately. That is what most mutations lead to. Now, there's a lot of other genetic disorders that are also connected to genetic mutations. So what is a mutation? Well, to mutate just means to change. That's what the word mutate means. So what is a genetic mutation? It's a change to your DNA. So our cells do a very good job of protecting our DNA. The DNA stays inside the nucleus. That's why we have that whole RNA process. So that way the DNA does not get exposed to anything if we can avoid it, right? But sometimes it does. Sometimes things get into the cell and cells are exposed to environmental factors that are going to cause changes to that DNA. And it's going to just copy wrong, or it's going to change one base or two bases or more. And it can happen also randomly. It doesn't have to be from something outside of the body. So mutations actually do happen a lot, but it's any change or mistake in the DNA sequence. Sometimes mutations are excellent. Sometimes mutations cause a species to evolve, and we get something like the human brain, right? That was a product of a mutation. Or you can lead to something like cancer, which is not a good one. So we're going to talk about the three major types of mutations in this lecture. The first type is called a point mutation. So think of the word point. How many people can you point at, to at once? Really, you can only point at one person, right? So if I'm pointing at you, it's one person. So a point mutation is very simply a change to a single base pair. So there's 3 billion base pairs in a strand of DNA. So a point mutation is a change to just one of those base pairs. And a lot of times these are what they call silent mutations because they don't even really affect the outcome of that protein, or sometimes they could be very dangerous. Like, for example, sickle cell anemia is a very, unfortunately, common genetic disorder, which is very dangerous, and that is caused by a point mutation. So what is a point mutation? So let's say Mr. Adolf is really cool is a sentence that is coded for by this strand of DNA. So we have ATG, TCA, CGA. And in some weird world, that codes for Mr. Adolf is real cool, right? So let's say I change one letter. And in this case, instead of it being CGA, it's GGA. That C, that cytosine, becomes a guanine. Well, is that going to impact the meaning of that phrase? Well, yes, it will. Mr. Adolf is well, you can fill in the blank for that one. So let me just explain it a little bit better with an example. So I have here a strand of DNA, and I've already done the work, so I didn't want you to have to watch me do it, but here we have a strand of DNA, and it's TAC, CCG, GAT, AAA, GTA, ACT. And what you end up with is it transfers to mRNA, that's our transcription, and then it translates to our amino acid chain. Now let's say I want to have a point mutation. Now, a point mutation would affect one single base pair. So let's say instead of this C, this G here, it's a C. So now that third set of bases here, that third codon, goes CCC. So instead of it being GGC, it's now GGG. So if I look here, G, G, G gets me glycine. Okay, that gets me glycine right here. Now, what did it get me before? Well, it was GGC. It also got me glycine. So I changed one base, but it didn't even affect the protein because these amino acids have multiple codes for them. So sometimes it doesn't have an impact at all. But let's say it was CCA instead of CCC. Well, now we're going to have GGU. So again, same scenario, it doesn't affect the protein. So a lot of times these point mutations are completely harmless. I'm going to give you an analogy because you know I love analogies. So let's say this is the lunch line. This is the line of people that are waiting to get their lunch. And everybody's hungry and there's only 25 minutes to eat. And you've seen the lines in the lunchroom before. They get really bad. Let's say somebody left the line, right? And somebody replaced them. Does anybody really care in that line? No, right? Nobody moved. There was no change to the number of people that are there. So overall, everybody's still happy. So that would be an example of a point mutation. One base changes and it could affect the strand. You could get a scenario where, let's say, instead of it being C, maybe the first letter gets changed. So maybe now it's ACC. Well, now we have a totally different amino acid. So it depends on the type of amino acid. The second type of mutation is called a frame shift mutation, and these are much more serious. So think of the word frame shift. The entire frame shifts, so everything moves. So a frame shift mutation is when there's an entire shift in a genetic message. That's a lot more serious. So I'm going to use an analogy here because you know I love them. So let's say you had a book and somebody changed one letter of one word. It's probably not going to affect the book that much. You probably won't even notice that there was a mistake. It probably happens. You probably have read books before where there's one letter wrong and it doesn't make a huge impact. So let's say though a word got added and everything got shifted over and now all the letters are in the wrong spots. That would be a frame shift mutation. It would affect a huge area of the book and you would have a much bigger impact. So this causes an entire shift to a genetic message, and there's two types. The first type is called a deletion. So if this is the original strand of DNA, if you remove one letter, if you pull one letter, everything shifts over. So if you take a look at the original strand here, it goes ATG, ACG, GCA, and so on, right? Well, now we remove that C, and when we remove that C, everything moved over because now our codons are in different spots. So now everything after that point becomes different, and you're going to get a much bigger impact from that mutation. The second type is called an insertion. An insertion is when you add a base. So let's say here is a perfect example of that. So it goes ATG, ACG, GCA. Well, now we're throwing a T in here. So we threw a T in where that G is, and everything moved down one. So now every single codon after that is affected. So I'm going to just show you an example of that, and that's pretty much what we're probably going to get through for this lecture for today. But let me just explain that. So if I go back to my example here, and I go back to my line example. So let's say now we have our scenario where these people are waiting in line for their lunch. And the first scenario where it was a point mutation, somebody just switched out. So nobody cared, right? Everybody was fine with it because nobody actually moved and nobody got affected. But let's say instead of that, this person just jumps into line and nobody switches. And now everybody has to move back one, right? All these people have to move down one. And now everybody behind this person is mad because now they have to wait longer in line, right? So this would be something called a frame shift mutation. So let me give you the example I'm going to use here. So here I have a strand of DNA, and let's say I want to make a frame shift mutation. So I add a base. It's not just a point mutation. I'm actually adding a base. So I'm going to go with my original scenario. So in my original scenario, if I draw my codon lines, right, like so, right, I have my 3 here, 3 here. So this has 1, 2, 3, 4, 5 bases, right? 5 codons, I should say. So let's say I add a base, though. I want to add one. And I'm going to add, it doesn't matter what I add. Let's say I add a C right here. Let me move these down, though. So now I'm going to add an extra C right here. Well, I just changed everything because now these codon lines are in the wrong spots, right? So now this codon is going to go 3 here, 3 here. Now these are going to be 3. This is going to be another 3, and now I just added one line here. So now everything after that point is going to be affected. So these amino acids are going to be the same here. These two are not going to be affected. But everything after that point is going to have an impact. And that is important as well. Where the frame shift occurs is going to matter. So let's say the mutation occurred at the beginning of the strand. Like way up here. Somebody jumped into the line up here. Everybody behind that person is going to be mad. But let's say the person just got in at the end of the line. Well, now it's not going to have as big of an impact. So if the person jumps in line at the beginning, everybody after it is going to be upset. If the person jumps in at the end, nobody's going to be upset. So where the mutation occurs matters. So I put the mutation right here. Well, let's say I put it here. Everything after it would be affected. So the farther along in that chain the mutation occurs, the less of an impact it has. So the more amino acids that get affected by this mutation, the bigger the impact. So in a frame shift mutation, if you look here, the entire protein sequence is changed. So if I compare frame shift to point mutations, frame shift are usually much more dangerous, and that's because the entire strand of DNA gets affected. Because when you add a base or you take one away, everything has to shift. Everything either has to move up one or down one. And if they all move up one or down one, every single codon is going to change after that mutation. Now, in a point mutation, it's just going to be the one. So we're going to finish up with the last type of mutation in another lecture. This is enough for one.